Alright, in meiosis, in contrast to mitosis, we are generating the gametes, or the sex cells. So whereas mitosis generated somatic cells, diploid cells, right, just by sort of copying them, in this case, meiosis is going to generate haploid cells. So what we're thinking of is that we're going to try and generate the gametes, okay? And so this would be sperm in the male and egg in the female. And these cells are 1N or haploid. Okay? So what we have to do is we have to think about this idea that we're going to start with a diploid cell, right? And at the end of the day, right, through a series of steps, right, we are going to end up with 4 cells, right? So if we think of them as 4 here, all of which are haploid. So all of these cells are haploid. Okay. And so we're going to do this then by 2 cell divisions. All right, so there will be 2 cell divisions in our process. Okay, so let's have a look. All right, so some terminology that we need to get started. Right, we'll remember that sort of this waist portion where these cells are, where the chromosomes are linked together is called the centromere. All right. A pair of chromosomes. Okay, so that's a pair of chromosomes that are the same in terms of the type of information they carry. So they are the same type. All right. So this would be homologous chromosomes. And we call this a homologous pair. All right. This red one here and this red one here, these are called non-sister chromatids. Now, we call them non-sister chromatids because they are not chromatids of the same chromosome, even though they are homologs to each other. So the sister chromatids have to be like this. So if we look at these two here, right, these are sister chromatids. And this structure containing 2 chromatids, right, is a single chromosome. All right. Now, the other thing we want to think about is we want to think about this cell. Okay, and so let me clean it up. Let me get rid of all of our stuff here. All right. There we go. Get it all cleaned up. Okay, so what we want to think about is how many chromosomes do we have? So we've got red ones are maternal. Blue ones are paternal. Okay. Now, the question is, what is N? All right, which N is the number of types of chromosomes. So N in this case is 3, right? Which means, if we notice, we've got 1, 2, 3 types. All right, so we've got the small, medium, and the large, right? And in this case, we actually have 1, 2, 3, right? So we have 2 sets of our 3 chromosomes, one of which are maternal and one set which is paternal. All right, so n equals 3, or diploid 2n is equal to 6. All right, so this will help us as we move forward. So the question becomes, what's going to happen in meiosis, right? This cell division where we're going to reduce the number of chromosomes. So if we look at this here, we have a pair of homologous chromosomes in our first cell. All right, so that's just a pair of homologs. Okay, and so I'm just going to call it a pair. All right, they are going to duplicate, right, which is going to give us 2 sister chromatids per chromosome. So notice we still just have a pair, all right, of chromosomes. Now, these things are going to divide. So if we look here, this purple line is meiosis 1, all right, and the second purple line is meiosis 2, all right, so that's our second division. Now, the key here is we refer to these two combined, right? If you remember back to the previous slide, this is a homologous pair. All right? So our homologous pair is going to be separated in meiosis 1. So we'll notice our paternal chromosome ends up over here in this cell, and our maternal chromosome ends up over here in this. So we separate the homologous pair. Okay? We separate the homologous chromosomes. Now, this cell has 1 chromosome. This cell has 1 chromosome and is 1N. This cell that we started with, right, is 2N. All right? Now, at this point, okay, so I just wanted to clean that up so you get a good look at this cell here as being duplicate. Oops. All right, I don't want that. Okay, so this thing is a duplicate. That there are 2 copies of our chromosome, 1 maternal, 1 paternal. That's diploid, 2N. Then in meiosis 1, we're going to pull the paternal chromosome over into this cell, and it is haploid now. There's only 1 chromosome present. All right, and there's 1 here. Rather than having 2 copies. Now, in meiosis 2, we are going to separate chromatids. All right? Okay, so our chromatids separate. All right? As opposed to chromosomes, which separate in meiosis 1. All right? So that's really the key. We end up with 4 haploid cells. All right? So we've got 1, 2, 3, 4 cells down here, and they are N, right? They are 1N. Okay. All right, so let's look at meiosis. So this picture shows us meiosis 1. And here we are going to separate homologous chromosomes, okay? So we have a prophase, we have a metaphase, all right, I'm just going to call that meta. Then we have an anaphase, which I'm just going to call ana, all right? And we have a telophase and cytokinesis. Okay. And of course, this is prophase 1, metaphase 1, anaphase 1, telophase 1. Okay? Now, if we look at prophase 1, there's a number of things to first kind of observe. The question is, what is N? All right? So we have a small chromosome, a medium chromosome, and a large one. So N is equal to 3. All right? we have both maternal and paternal versions of each of those. So we have a maternal and a paternal long one, a maternal and a paternal medium, and a maternal and paternal short. So we are really diploid, right? 2n equals 6. And so this cell is 2n. Okay? Now, there's a few things we want to look at, all right? And number one is that we have homologous chromosome pairs. Okay, so we have homologs. Okay, so we're talking about homologous chromosomes. And in this phase, right, in meiosis 1, all right, what our goal is to separate the homologous chromosomes from each other. All right, so we've got homologous chromosomes, and that's what each of these pairs is. Okay. Now, while we are in prophase, we'll notice that we're starting to see the spindle form. All right. We've got crossing over occurring. All right, and so this is that circle is a crossover, and we call that a chiasmata. All right, spelled chiasmata. All right, so we have those at the crossover points, and it is between non-sister chromatids. Right, so basically we have the a chromatid from the paternal chromosome, the blue, and a chromatid from the maternal chromosome in the red, in the crossover. All right. All right. Now, from here we enter metaphase. Now, in metaphase, what you will notice is that the homologous pairs line up together. All right. So the homologous pairs line up together. Okay. So the homologous pairs line up, all right? The other thing we want to notice is that the spindle apparatus, right, is binding to one sister chromatid of the combined pair of chromatids of the maternal chromosome, and one spindle is attached to the paternal chromosome, all right? So we're not separating the two chromatids from each other, we're separating the two homologous chromosomes from each other. All right, and we can see this in each of these instances. Okay. Now, the other thing I want you to look at here is that we've got a paternal on this side, a paternal on this side, and a paternal on this side. And the way that the homologs line up is totally independent. So how this chromosome aligns is independent from how this chromosome aligns, and both of those that have circles around them line up independently from the one in the middle, meaning that the maternals don't have to line up so that they all end up in the same cell, all right, and the paternals don't have to line up so they all end up in the same cell. So they're going to basically randomly align, okay, and we call this independent assortment. Okay, so this is setting up independent assortment. Okay, so I'm just going to abbreviate that, independent assortment. Okay, so it's setting that up. Okay? Alright, let's get rid of some of that junk. Alright, now anaphase is where we're going to pull the homologous pair of chromosomes apart. So homolog, if we look at these here, right? So here's a homolog and here's a homolog, right? And this one is heading towards this pole and this one is heading towards this pole, and we have separated them. You will notice that each chromosome exists of two chromatids. So this one, for example, has two maternal chromatids, right? Because it's the maternal chromosome. So it has two maternal chromatids. All right. This one is still two chromatids. All right. Everybody good with that? So we've got that. So now what we've done is we have separated out the homologs. So we'll notice then in telophase, where we're going to split these two cells, right, in telophase and cytokinesis. So if we think of this as having a division here, now we'll notice that we have 3 chromosomes here and 3 chromosomes here. Okay? Notice if we go back to the prophase cell, we have 1, 2, 3, 4, 5, 6 chromosomes, right? 6 total. And that is the 2n number. N is equal to 3 for my prophase cell. So here, since I have 3 chromosome, I am at N, or haploid. Because I have 3 for this cell, I am at N, or haploid. All right, so we have already reduced, and so we call this the reductionist phase, and we are reducing the total number of chromosomes in our cells. Okay? All right. So now if we move to meiosis 2, all right, so we have a prophase again. All right, so we have prophase 2, we have metaphase 2, anaphase 2, and telophase cytokinesis 2. All right, so in this case, all right, prophase 2, this cell is haploid. All right, each of these cells are haploid already. We have 6 sister chromatids in there and 3 chromosomes in each of these cells. So we're going to rebuild our spindle apparatus, which we can see occurring here. All right. We also, all right, you can also see clearly on the chromosomes that crossing over has occurred, and we have recombinant chromosomes. Okay. And actually at this point it's just one chromatid of each chromosome has the recombinant nature. Okay? So as we move to metaphase, and let's just look at the top, all right, our 3 chromosomes line up at the metaphase plate. This time, each sister chromatid has a kinetochore. And so this chromatid will be pulled in this direction, this chromatid will be pulled in this direction. This one will go like so, this one will go like so, and so forth. So now we have the ability to separate sister chromatids into chromosomes. And so if we look at anaphase here, now we have chromosomes. All right, so we have chromosomes. Boy, that's ugly. Let's clean that up. All right, so now we are at chromosomes. Instead of here, we have 3 chromosomes. All right, and we have 3 chroma, and I'm just going to call them TIDs. All right, T-I-D-S, TIDs, for the chromatids. All right, so whoops, sorry, we don't have 3, we have 6. Sorry, so don't let me mess you up. All right, so we have 6 chromatids. All right, 3 chromosomes. As we split this, now we're going to end up with, after telophase and cytokinesis are completed, we will end up with 4 cells, top and bottom, all right, each with 3 chromosomes, 3, 3, 3, and 3, which is our haploid number. So we end up with 4 haploid cells, okay? Now, when you go through this, you want to really be able to think about the numbers of chromatids, the numbers of chromosomes, and when you in anaphase, right, when you go through anaphase, you separate those chromatids and they become chromosomes, and that's part of the key to making this whole thing work, okay? All right, so let's compare mitosis and meiosis. So can you tell which side is which? Well, the thing I do to look and tell the difference is if you look at prophase, all right, so this is prophase here, okay, and it's pro over here as well, all right. What you want to look at is are the chromosomes, right, they've been duplicated, so each chromosome consists of 2 chromatids, but are they bunched up in homologous pairs? That occurs over here. Notice how they're bunched up in homologous pairs, and over here they're independent. And that basically is a dead giveaway that this is mitosis, and this is meiosis. All right? Now, so we have this pairing up in meiosis, we don't. Then if we look at metaphase, okay, and it's metaphase in mitosis, and it's metaphase 1 in meiosis. Notice all the chromosomes are lined up in a single file line in metaphase. So we've got this sort of single file line here. In contrast, in metaphase 1 of the meiosis, we actually have 2 lines of chromosomes next to each other, right? Because we're in homologous pairs. The other thing we'll notice is that the maternal and the paternal chromosomes are independent. They don't have to align together. So we don't end up with all the paternal chromosomes on one side and all the maternal chromosomes on one side. Okay? Then we go through anaphase. All right, and in this case, we're combining anaphase and telophase. Okay? So here's anaphase and telophase over here, and these are, of course, anaphase, telophase 1. So if we look at meiosis and mitosis, so if we look at the mitosis, here we separate chromatids, all right, into chromosomes, and this cell is 2N, this cell is 2N, this cell is 2N, okay? If we look at meiosis, we separate chromosomes, all right? So we have 2 daughter cells of meiosis 1, and we have 3 chromosomes in this cell here, okay? And we have 3 chromosomes here. These cells are haploid, all right? We start out with 6 chromosomes up here, diploid. After anaphase 1, we are haploid, all right? Then after meiosis 2, all right, so M2, meiosis 2, we separate the sister chromatids, and we end up with 4 haploid cells, all right? Okay, so if we look at this then, some of the key things are that we have basically divided our cell, right, in terms of meiosis. We have reduced the total number of chromosomes from 2N to 1N, all right? Now, the next thing we want to talk about is how the genetic variation is created, all right? So we want to really look at variation and how we generate the variation through sexual reproduction. All right? So, first of all, we've got this mutation, which gives us all of our variation, okay? Or the working material, all right? So it is the original source of genetic diversity. That's what mutation is, okay? Allows us to create different versions of genes that we call alleles. In sexual reproduction, so if we think about this as generating alleles, right, which are our copies of genes, right, different versions of genes, okay, and then what sexual reproduction allows us to do is to shuffle up the alleles, okay, make new combinations of alleles, okay. So let's look at this, all right. So first of all, let's see how many different combinations we can make of chromosomes, all right? So in the human, all right, we have 23 chromosomes, which means that we have 2 to the 23rd possibilities in terms of total chromosome combinations. This is just in the chromosomes, okay? Now, in this case, we have N equals what? So you should think that through. How many different types of chromosomes are there? And you should have answered 2, all right? And so in this case, 2 to the N is equal to 4, all right? So what we want to do then is think about how many ways can we do this. So if we have possibility 1 here and possibility 2 here in terms of how the chromosomes align at the metaphase plate in meiosis 1, all right, we can have both paternals on one side, both maternals, and we end up with 4 cells down here at the bottom at the end of this meiosis, all right. We end up with 4, but we only have 2 combinations. So we've got 1 possibility here, 1 possibility here. Our other option is that 1 maternal goes with 1 paternal, and 1 maternal goes with 1 paternal. And that will give us possibilities 3 and 4. All right? So what we're seeing then is that we can end up with different combinations of chromosomes, right? So we've got 4 possible, and I'm just going to refer to these as combos, all right? So we've got 4 possible combinations of chromosomes, all right? Now, the other thing that occurs is that we get recombinant chromosomes through crossing over, right? This will create variation as well. And so we're going to look at crossing over, all right? And the goal is that we're at the end, if we look at these two down here, all right, these are what we call recombinant, all right, recombinant chromosomes. All right, so if we look at this, then we have prophase 1, and that is where we get the crossing over, and we create these recombinant non-sister chromatids. All right, and so this is anaphase 1. All right, so we've separated the homologous pair. All right, and when we go into anaphase 2, all right, we now separate the sister chromatids, only you'll notice the sister chromatids are no longer identical to each other, right? So if we take these two sisters, okay, so those are our sister chromatids, they are no longer identical to each other. And that increases the total amount of variation that we can have. All right? So the third way that we can have that sexual reproduction will lead to variation is random fertilization. Okay. So in humans, this means that you take one, right, of our 7.4 million possibilities, or 8.4 million possibilities, right? So you take one of 8.4 million, okay, and you're going to take one of 8.4 million eggs, right, possibilities in terms of the combination of those eggs. So this one up here, right, this is my sperm, all right, and down here I have my happy little egg. Okay, so when you randomly pick the sperm, you randomly pick the egg, and when you bring these two things together into a zygote, you have one of 70 trillion possibilities. Incredible chance there that you actually create this thing. So that's random fertilization. It's only made possible by the fact that we have these haploid cells, right? If we didn't have haploid cells, we couldn't unite them together and create our organism. All right, so that's the basics of meiosis, and keep plugging away at it.